Welcome to this lecture on evolutionary biology. Today, we are going to explore a fascinating and crucial concept known as the bottleneck phenomenon. We will discover how sudden, catastrophic events can drastically alter the genetic makeup of a population, and why understanding this is so important for the survival of species on our planet. Before we dive into the formal biology, let us imagine a large glass jar filled with thousands of jelly beans of 10 different colors. This jar represents a highly diverse population of animals. Suddenly, a disaster strikes. You accidentally knock the jar over, spilling almost all the jelly beans into a fast-moving river where they are lost forever. You manage to save just 5 jelly beans that happen to be stuck near the narrow neck of the jar. By pure chance, 4 of these are red and 1 is blue. The other 8 beautiful colors are completely gone. If these 5 beans start a new jar, it will lack the rich diversity it once had. This perfectly captures the bottleneck phenomenon. Now that we have our jelly bean analogy in mind, let us look at this visual representation of the bottleneck effect. On the left, you can see the original population, represented by a large flask filled with many different colors, indicating high genetic diversity. Then we have the bottleneck event in the middle. Notice how the drastic population reduction forces only a tiny, random sample through the narrow neck of the bottle. Finally, on the right, we see the surviving population. It is much smaller, and more importantly, it has lost almost all of its original color variations. The new population is genetically uniform, simply because of the random chance of who survived the event. Moving from our analogy to formal biology, what exactly is a population bottleneck? In evolutionary biology, it is defined as a sharp, drastic reduction in the size of a population. This is usually caused by severe environmental events, such as earthquakes, floods, extreme droughts, or even human activities like overhunting and habitat destruction. When a population shrinks this drastically, the few surviving individuals only carry a very small fraction of the genetic diversity that existed in the original larger population. It is a catastrophic event that instantly wipes out generations of genetic variation. Understanding the cause of the reduction leads us to a critical point about how evolution works during a bottleneck. The individuals who survive these disasters do not survive because they have better genes or higher fitness. They survive purely by random chance. Because of this, the bottleneck phenomenon is a powerful form of genetic drift. As the population eventually recovers and grows in numbers again, its genetic composition will only reflect the alleles, or gene variants, of those few lucky survivors. Consequently, the new population has significantly reduced genetic variation, which can permanently alter its evolutionary trajectory moving forward. So, why does this loss of variation matter so much? Understanding the bottleneck phenomenon is absolutely crucial because genetic diversity is the raw material for evolution. It is exactly what allows a population to adapt to new diseases, climate changes, or predators. When a bottleneck wipes out this diversity, the population loses its evolutionary flexibility and becomes dangerously uniform. A classic real-world example is the modern cheetah. Around 10,000 years ago, during the last ice age, cheetahs went through a severe bottleneck. Today, they are so genetically similar to one another that they suffer from poor reproductive success and are highly susceptible to diseases. Now that we see the real-world dangers, how do biologists actually measure this loss? To understand the bottleneck phenomenon mathematically, biologists use a metric called heterozygosity. You can think of heterozygosity as a numerical score of genetic diversity. Specifically, it represents the probability that two individuals randomly chosen from the population will have different versions of a gene. When a bottleneck occurs and the population size drops to a very small number, we can calculate exactly how much of this genetic diversity is lost per generation. This allows conservationists to predict the future health of an endangered species. This brings us to the mathematical representation of genetic loss. The formula you see here predicts the remaining heterozygosity after a certain number of generations, and it is a fundamental equation in population genetics. Let us break down the symbols. H sub t represents the expected genetic diversity after a specific number of generations have passed. H sub 0 stands for the initial diversity before the disaster struck. The variable n represents the effective population size, which is the number of breeding individuals that survived. Finally, t is the number of generations the population remains small. Notice the fraction 1 over 2n. This represents the rate at which diversity is lost. If n is very small, this fraction becomes large, meaning a huge chunk of diversity is subtracted away. Let us apply this formula to a concrete example. Imagine a thriving population of rare birds with an initial heterozygosity of 0.80. A hurricane leaves only a tiny bottleneck population of 5 breeding birds. We want to find the expected diversity after just one generation. We substitute our known values into the equation. Initial diversity is 0.80, n is 5, and t is 1. The denominator of our fraction becomes 2 times 5, which is 10. This means the rate of loss is 1/10, or 10% per generation. Subtracting this from 1 leaves us with a retention factor of 0.90. Multiplying our initial diversity by this factor gives a final heterozygosity of 0.72. Physically, this means the hurricane caused an immediate, permanent loss of 10% of the species' genetic variation. Having seen the impact of a single generation, we must now consider what happens over longer periods. In reality, endangered species often stay at small population sizes for many years. When a population remains small, the loss of diversity does not just happen once, it happens repeatedly, generation after generation. This creates a compounding effect, much like compound interest in a bank, but in this case, it is a compounding loss of genetic health. This compounding effect demonstrates exactly why it is critical for conservationists to intervene and grow endangered populations as quickly as possible, before the genetic variation bleeds away completely. Let us calculate this compounding effect with a second example. Suppose a population of wolves is hunted down to just 10 individuals. Their initial heterozygosity was 0.60. We want to know the diversity if they remain this small for 5 generations. We plug in n equals 10 and t equals 5. The fraction inside the parentheses becomes 1 over 20, or 0.05. This means they lose 5% of their diversity every single generation. The retention factor is 0.95. Because this loss compounds, we must raise 0.95 to the power of 5, giving us approximately 0.7738. Multiplying this by the initial diversity gives a final heterozygosity of 0.464. The prolonged bottleneck has severely depleted their gene pool, making recovery incredibly difficult. We have covered a lot of ground today, so let us summarize the most important ideas. First, The bottleneck phenomenon is a dramatic event that shrinks a population and randomly strips away its genetic diversity. Second, because survival is based on luck rather than fitness, it acts as a powerful mechanism of genetic drift. Third, as our mathematical models proved, smaller bottleneck sizes and longer durations at those small sizes lead to devastating, compounding losses in heterozygosity. Ultimately, understanding this concept helps us realize why preserving large, diverse populations is the absolute only way to ensure a species can adapt and survive in our ever-changing world. You now have a solid understanding of the bottleneck phenomenon.